Hello and welcome to another episode of Literature Talk. Today we have Anubhav Ghoshal with us. He completed his BSc from Banaras Indian University, Varanasi, and is currently a third year integrated PhD student in the Department of Organic Chemistry at the Indian Institute of Science, Bangalore. In today's talk, he will present DNA copper hybrid catalysis, unlocking a toolbox for enantioselective synthesis. So, Anubhav, welcome to the talk and the screen is yours.
All right. Thank you, Mahesh, for such a wonderful introduction. Greetings, everyone. It's good to be here, and thank you for this opportunity. So, the topic of today's discussion is DNA copper hybrid catalysis, unlocking a toolbox for enantioselective synthesis. I shall begin with a brief introduction to DNA and its structural and functional aspects, followed by the design and synthesis of the catalyst and its application in organic synthesis, some of the common organic transformations. Then we shall move on to the recent advances, some future perspectives, and of course, a few concluding remarks. Now, from a chemist's point of view, or rather a structural perspective, DNA, the molecule of life, may be defined as a pair of polynucleotide sequences that tend to run anti-parallel to each other and are connected in terms of hydrogen bonding interaction between the nitrogenous bases, which are further held intact to the sugar phosphate backbone by means of an N-glycosylic linkage. Now, conformationally speaking, DNA may be present in several forms such as A, B, and Z that may be either right-handed or left-handed in correspondence to a fixed axis of rotation, thus imparting it a very special form of chirality known as helicity. Interestingly enough, this chirality or helicity of the DNA can indeed be transferred into molecules that are covalently bound to it. This was shown in the fundamental works of David R. Liu at University of Harvard when he conjugated two reacting substrates and subjected them to a nucleophilic substitution reaction, thereby rendering a transfer of enantioselectivity or transfer of chirality of the DNA into those substrates as well as the subsequent product. Although these components never saw a complete chromatographic purification, most of the data was analyzed through mass spectrometry, multi-top mass spectrometry, and the chirality, of course, by a chiral HPLC column. However, it led to a very basic foundation to the fact that the chirality transfer can happen in case of DNA. And that thought or that idea makes one wonder if DNA can be used as a catalyst for enantioselective organic synthesis. Now, to bring that to reality, a young Gerard Roelfs, along with his supervisor Ben Feringa at the University of Groningen, decided to devise a catalyst of their own, composed of a DNA biopolymer, which was intercalated with acridine-based ligands, followed by a copper source. It was chelated to a copper source, and finally, when it was subjected to a Diels-Alder reaction between a cyclopentadiene and an azachalcone, it resulted in an adduct which was formed majorly in endo. Upon iterating through the linker lengths as well as the side chain substitutions, it was discovered that the chirality of DNA that was transferred into the molecules or the product or as well as the substrates was indeed a function that was driven by the proximity of the reacting species to the DNA. However, you can say that enantioselectivity over here remained to be a major problem. Now, in order to deal with that, Royalfest decided to screen through a new set of intercalators which would decrease the space between the reacting species as well as the DNA polymer. So he relied on intercalators which are mostly composed of bipyridine as well as phenanthroline based moieties and experienced that the enantioselectivity was enhanced by a great margin, as well as there was a spike in the rate of the reaction by several folds. Now, mechanistically, the whole 4 plus 2 cycloaddition reaction that is driven by a Lewis acid catalyst can be segmented or fragmented into three sections, mainly the first being the binding of the copper DNA complex to the dienophile, followed by the binding of the diene to the activated dienophile, and finally the sweet release of the product from this seemingly complex set of catalytic system. Now, upon subjecting few of these kinetic as well as thermodynamic parameters to several experimentations, it was discovered that the presence of the DNA tends to favor the the step wherein the diene tends to react or interact with the activated dienophile complex and pushes the equilibrium in favor of the major enantiomer. Furthermore, the change in the thermodynamic parameters in the presence of the copper DNA system revealed a very special non-classical hydrophobic effect that actually corresponds to the pi-pi stacking interaction between the arene rings of the substrates as well as the DNA catalyst or the DNA backbone. Upon subjecting this system to several DNA sequence screening, it was discovered that the presence of GC-rich region in the DNA not only enhances the enantioselectivity, or you can say it not only enriches the enantioselectivity, but also triggers the rate of the reaction by a huge margin that is almost exponentially, which in return stabilizes the whole reaction microenvironment. Upon building such insights and fundamental understanding of the concept of the working of the DNA hybrid catalyst, this particular system, or you can say the second generation catalyst, the first generation being the acridine and the second generation being the dimethyl bipyridine system, saw its application in several reactive transformations, namely the Fiddlecraft alkylation reaction, which was well tolerated over a wide variety of substrates over several functionalizations in almost very good yield as well as enantioselectivity, followed by some molecular dynamic simulation, which reveal the binding states, the many binding states of the substrate with the DNA backbone throughout the course of the reaction. It further saw its application in a Michael addition reaction, which gave excellent yield as well as enantioselectivity for diverse functionalities as depicted here. Now, upon stereochemical studies to gain deeper insights into the working mechanisms, it was discovered that the DNA backbone tends to impede the approach of the nucleophile from the rephase, thus favoring a psi phase attack. The same system of catalyst saw its application over a myriad of reactions, namely synhydration, intramolecular Fiddlecraft alkylation, fluorination, and cyclopropanation, just to name a few, each with a considerable enantioselectivity. However, the seemingly robust system did come with a few setbacks, the major ones being moderate to poor enantioselectivity for most of the substrates, a limited library of catalyst, and of course substrate scope, requirement of higher equivalent of substrates, which varied somewhat around 100 to even 1,000 equivalents just to push the reaction towards completion. And most importantly, from a mechanistic point of view, the non-specific binding of the intercalator to the grooves of the DNA. So there was, of course, a requirement for a new system of catalyst, a new catalyst design or a new protocol, a synthetic approach for a better catalyst. And in order to do so, to change the nature of the binding from a non-covalent supramolecular interaction to a rather strong permanent covalent interaction, Royalfish in the earlier days uh, tried to employ the concept of phosphoramidite chemistry, wherein his group took a functionalized sequence of DNA, ligated it with a non-functionalized strand, and finally annealed it to a complementary sequence of both to generate the entire catalytic system, which only brought about a marginal improvement in the yield as well as enantioselectivity. Now, considering the highly sensitive and cumbersome synthesis of these phosphoramidite bonds, followed by the limited scope of modification, this approach essentially does not seem worth the bargain. Therefore, one might think of resorting to something that looks much more natural. That is something that happens within us, within every human, within almost every living being, and that is if mutation masked by evolution. Ladies and gentlemen, these nitrogenous bases present in the DNA backbone, such as cytosine, have the ability or the tendency to undergo mutation driven by certain enzymes, in this case, a cytosine deaminase, to trigger a deamination reaction that starts with the protonation of the amine, followed by several electron swaps to generate somewhat of an enolic species that automerizes to generate the uracil. Now, this uracil, if bound to the DNA phosphate backbone, or more like the sugar phosphate backbone, can undergo a base pair scission exactly at this N-glycosylic linkage in the presence of a glycosylase enzyme to generate a free aldehyde, which of course can tautomerize in a closed ring Furanose structure and its aldehyde free aldehyde form. Now, interestingly, this aldehyde group, the free aldehyde, is susceptible to several nucleophilic addition reactions, which can easily yield hydrogens and oximes that are not only hydrolytically stable but can be easily modified and are absolutely bioorthogonal in nature. Now, to bring this projection into reality, a group of scientists at the National University of Singapore, very recently, you can say, devise a strategy wherein they take this mutated strand of sequence of DNA and subject it to a scission in the N-glycosylic bond or the N-glycosylic linkage by means of the enzyme UDG, that is uracil DNA glycosylase, that is abundantly available in E. coli. Now, this E. coli being a bacteria that can easily be cultured in almost every laboratory to generate the free aldehyde species, which undergoes a nucleophilic addition followed by a condensation reaction to generate a massive library of 44 DNA-based catalysts, which can be selectively installed into the complex architecture of the DNA at several other positions as per the requirement. Now, few of these catalysts were subjected to screening by means of a Michael addition reaction, wherein the best performing was taken for further optimization in terms of the chiral microenvironment, uh, wherein a 17-mer hairpin DNA was taken and it was mutated at a particular site. Over here, L1 refers to, of course, the ligand or the modified bioconjugated catalyst system. And the reaction microenvironment essentially refers to its neighboring bases as well as the one at the opposite. Now, upon optimizing this, we got a catalyst which showed efficient loading as well as a high turnover number. So to put it to test, it was again subjected to some standard reaction, for example, a Michael addition reaction, wherein it brought about excellent yield, almost perfect conversion, and of course, enhanced enantio enrichment. Now, considering the Fiddlecraft conjugated addition reaction, which proceeds through an enantioselective protonation step, of this century, or you can say in the first 10 years of the first decade of this 2 decade long journey of this whole field. And you can see it surpasses its predecessor in terms of yield as well as enantioselectivity selectivity every single time for every single functional derivatization of the substrates. And the cherry on the top is that this catalyst helped us to achieve a substrate scope comprising of the first ever DNA catalyzed atrope selective compounds, again with exceptional yield as well as enantioselectivity. Now, with a thing that is so robust, you would of course be willing to take it up a notch, increase the difficulty, and try even complex reactions with a catalyst like this. So, to take things up a notch, the same group at NUS decided to take inspiration from DNA's innate or inherent ability to form ion pair interactions with proteins, peptides, and several other enzymes and biomolecules by virtue of their phosphate backbone and mimic this ability for ion pair asymmetric catalysis in DNA's innate domain, that is water. In order to do so, they took the same 17-mer hairpin DNA, uh, bioconjugated it with the same catalyst, which was screened out, and subjected it to a set of reactions which either included or proceeded through a cationic uh, species, namely a fluorination reaction, an intermolecular Mannich reaction, a photocross dehydrogenative coupling, and an intramolecular Mannich reaction. At first, the reaction microenvironment was optimized for each of the reaction. That is, the neighboring bases were adjusted or changed or swapped to get the best enantioselectivity and conversion, following which the substrate scope was generated, exhibiting again an excellent yield as well as enantioselectivity for most of the substrates, with several other functionalization and derivatization for each and every reaction. However, the most important thing that was supposed to be seen over here, or that was actually subjected to study, is whether is actually to discover the fact if the phosphate group indeed has some interaction or has a role to play in this form of catalysis. Either it takes part in the ion pair catalysis. So first they started with a molecular docking study wherein it, to their delight, it revealed the fact that few of the phosphate groups do interact with the substrate that is flanked between the grooves of the DNA. And to further acknowledge this in terms of experimentation, to gather several experimental evidences and insights to this, they subjected the DNA sequence to a scanning, wherein the phosphate groups in the backbone of the DNA was swapped with a phosphorothioate molecule, essentially changing or altering the electronegativity as well as the electron density of the backbone, of course, bringing several changes to the interaction or the intensity of interaction between the reacting species as well as our catalyst, and generated the reaction profile in terms of the enantioselectivity as well as the conversion for two of these reactions, that is fluorination and intermanic or the intermolecular Mannich reaction. And just considering a few specific PS sequences, namely the 10, 13, and 15, and so on, they subjected them to further modification with a neutral MP group, that is methyl phosphonate, which will supposed to again alter the interaction rates by hopefully several folds. And to our delight, it was observed that in doing so, the enantioselectivity as well as the conversion saw a great deterioration, thus confirming the absence of ion pair and the importance of DNA phosphate backbone in ion pair catalysis, or particularly, to be specific, ion pair asymmetric catalysis. Now, with a system that is so robust, that shows excellent yield as well as enantioselectivity over a wide variety of substrates through several reactive transformations, one can always gather the courage to test it further by employing it into, say, much complex reactive transformations such as multi-component reactions, as well as alpha CH functionalizations to impart enantioselectivity to the desired compounds, which are indeed of great biological importance to the human world. And of course, either in a near future or far future, we can always hope that a system as versatile as this does see its way into industry, or somehow, someday, somebody figures a way out to use the transformative abilities of the DNA hybrid catalyst into a system as small yet complex as a living cell. Now, of course, in the end, in case you enjoyed it or perhaps not, uh, we started our journey with basic structural and functional aspects of the DNA, followed by the transfer of chirality, the principle of transfer of chirality, as shown by David R. Liu's group, followed by the design of the first and second generation catalyst, wherein Gerhard Rolfes was mostly involved and saw some of the reactive transformation using those catalysts. Then we moved on to the bioorthogonal method or approach of synthesis of a library of catalysts. Then we tried to analyze or investigate the uh, importance of the phosphate backbones in ion pair catalysis, and finally had a sneak peek into the future and how it actually looks like or what it could hold. And with this, I would like to conclude and, of course, like to thank you all for your kind attendance.
Nice presentation. If you enjoyed this session, don't forget to like, share, and subscribe. We'll meet you in the next episode, next session. Thanks for watching.